So now let's continue. Yeah. So we went over sensory motor stage in the Piaget's moral stages of cognitive development, not moral. For moral development, it is Kohlberg. We're gonna probably cover that somewhere else. Um, uh, so the first stage is the sensory motor stage. Um, it goes from 0 to 2 years old, and it is the end of the sensory motor stage, where children likely have object permanence. And object permanence is like peekaboo, like when you kind of you kind of kind of hide an object, and when it goes out of sight, the child realizes that. So that's at the end of 2 years old, 2 years age. The child realizes that the object is not disappeared, and children are quickly learning about and making sense of the new world around them. So this is mostly done in sensory. Think a child putting all their toys to their mouths, and motor, a child crawling everywhere styles, whatever that means. So it should be that children are quickly learning about and making sense of the new world around them. Okay, so this is the characteristics of the sensory motor stage. They're quickly learning about the environment around them, and this is mostly done in sensory. Think a child putting all their toys to their mouths. Yeah, so they just want to touch things, and whatever they see, they're curious about it, right? And also motor. The motor part relates to a child crawling everywhere, right? And then the next stage is the pre-operational stage. It goes from 2 to 7 years of age, and the milestones achieved for the MCAT purpose. There isn't a strong milestone for this stage. Interesting. I actually doubt that, because there were milestones recommended by the Kaplan books, but we're gonna see. The characteristics are that thoughts are symbolic. Children are starting to use syntax and grammar to express full concepts, and their imagination and intuition are strong. So they make they think in symbols and syntax. What is a syntax? So let's see. We have Piaget's. It's not related here. It's more about the language development models, but we're going to use that later. So for language development models, you need to know what is a syntax. So yeah, the main thing is that from 2 to 7 years old, in the pre-operational stage, the children start to think in symbols and syntax to express full concepts. And object permanence, yeah, think of peekaboo. So object permanence is the ability to understand that objects do not disappear just because they go out of sight. That is a good way to put it. So objects do not disappear because they go out of sight. And there's the formal operational stage, and it is from 11 plus years of age. and the milestone achieved. Again, here it says there isn't a strong milestone for this stage, but the characteristics are that theoretical thinking has begun. So we can here ask Piaget's cognitive development model, and we have here that what are the strong milestones achieved at the end of the, so what's after the sensory motor stage? There's the pre-operational stage, and the formal operational, right? And formal operational stages. So here in the formal operational stage, the theoretical thinking has begun. There is an abundance of abstract logic and reasoning. Strategy and planning is possible, and the child has started to apply concepts across each other. Correct? Right. So the children start to apply concepts when they're over 11 years old. Usually this means that they are past the primary school, so they have abstract logic and reasoning and strategy and planning, and that's possible in this age, and they apply concepts across each other. And we have the conservation, which is the understanding that matter is conserved regardless of the container or its shape. Oh, this is one of the milestones for one of the ages. I'm guessing it's for 7 to 11 years old. We will see. So it should be for the formal operational stage, if I'm not, or the concrete operational stage. Yeah. So conservation is the milestone achieved for 7 to 11, the concrete operational stage. And the characteristics is that concepts are attached to concrete situations. So time, space, and quantity are understood and applicable in concrete situations. Conservation is the understanding that matter is conserved regardless of the container or its shape. And let me just set a timer for a second. We have the 1 hour timer, and we're going to put our devices on do not disturb mode. And then we have the concepts that are attached to concrete situations: time, space, and quantity. They are understood and applicable in concrete situations. So conservation is, knowing that the matter stays the same regardless of changing shape or size of the container. And so 0 to 2 years old, we have the sensory motor stage. 2 to 7 years old, we have the pre-operational stage. And then we have the concrete operational, and then we have the formal operational. So the conservation is frequently illustrated by pouring a tall and skinny glass of water into a shorter and fatter glass. There is a YouTube video on this, so if anyone's interested, you can watch that. And a child who has mastered conservation will realize that the amount of water is identical between the two glasses. However, a child who does not have that milestone mastered will say the tall, skinny glass has more water in it because it's just taller, right? And that is a milestone that is achieved after 7 years old. So children after 7 years old should be able to say, no, the amount of water is the same in both of those containers, regardless of the shape. And yeah, so for formal operational and pre-operational, we need to know what milestones there are, if there isn't any, as stated here. Okay, cool. Let's go to the protein structure. So what structure is the local three-dimensional arrangements that a protein contorts into due to the backbone of the amino acids having chemical interactions? So if the backbones of the amino acid are having chemical interactions, it is a secondary structure thing, right? Because the backbone is related to the secondary structure. It's mainly the hydrogen bonds that take place at the backbone of the protein, the amino acids. And then what structure is the chain of amino acids? So it is the primary structure that has the chain of amino acids. And if you have a point mutation, for instance, that point mutation could affect tertiary and secondary structure, but by definition, it disrupts primary structure. So we should keep in mind here that point mutation can affect tertiary and secondary structure, but the primarily, by definition, it disrupts the primary structure because in point mutation, let's take a look at point mutation. Point mutation. So if this is our DNA and our codon, we read it from we create the anticodon from the 3' to 5'. I believe we can check that later. And then we have the mRNA, which is the same as the codon on the DNA strand. And then there's a protein that is specifically translated by that codon. And in point mutation, these are the different mutations explained here. In point mutation, it's only one base that is changed. So the AAG is point mutated to AAA, right? And if the AAG, if the G is mutated to A, it results in a silent mutation, right? Because they both give a lysine protein. They both translate for lysine protein, right? But if the first amino base, the first one, which is A, is point mutated to T, you have a stop codon instead, because TAG is UAG in mRNA, and it turns into a stop codon. and it is nonsense. It is non-synonymous. So all of the other ones, because they give it the translate for a different protein, they are non-synonymous, and this specifically here is nonsense because non is related to stop codon. So whenever you see a non-sense, it's when it's nonsense, it doesn't make any sense. So it's stopped because it's stopped, right? And then the middle one, if AAG is turned into is point mutated to AGG, then you can see the AGG translates for arginine. Now, arginine is a conservative. In a sense, we need to understand also what is the difference between conservative and non-conservative. So if it's conservative, I think it's because they are both basic, but just because this one is polar, it's a different side chain. You can see that threonine is a non-conservative missense mutation. But anyways, back to our point that point mutations are you see, so corresponds to amino acid leucine. So because this is leucine, it's a silence mutation. It's synonymous. It's silence. We have also a frame shift mutation. Every amino acid that follows will be altered, so it changes the whole frame. The valine is a missense mutation, and nonsense because it's stop codon. Now, our point here was that point mutation is by definition disrupting the primary structure of the protein, of the chain of amino acids. Why? Because it is it is affecting the whole, like in the frame shift you can see that it can affect the rest of the proteins, right? The chain of the amino acid is affected, and the only exception is when it's a silence mutation, but still the base is different. So this C might be interacting, bonding with other bases or molecules by mistake. So that's my guess. Yeah, this one was a good one. We're going to put this here, and we can also put this one. I mean, yeah, sure, doesn't bother. Okay. Cool. Let's actually put it here. Now, the two most common types of secondary structure that are tested are beta sheets and alpha helices. Yes, this is easy peasy. The backbone of an amino acid consists of an amine group, a carboxyl group, and an alpha carbon and a hydrogen. This is the backbone of the amino acid. Yeah, the backbone means like the whole thing, main thing. And the tertiary structure is the geometric 3D folding of moieties due to side chain interactions to form an entire functional globular or structural protein. Ooh, I don't know what I'm reading. So, let's see. Tertiary structure is the geometric 3D folding of moieties. What's the meaning of moia? Like, I knew it. Okay. Let's see. So moieties are two parts or halves into which something is divided or split, often in equal or distinct portions in various contexts. Moieties can represent divisions within social groups, biological components, or chemical substances. In anthropology, moieties can refer to divisions within a society where each group is usually connected by kinship or ritual relationship. These divisions might have specific roles, responsibilities, or interactions within the larger community. In biology, moieties can denote distinct parts or components of a molecule, particularly in organic chemistry, where a molecule might be split into different functional groups or sections. The term is versatile, used in different fields to represent halves, divisions, or parts of something, often in a way that maintains a sense of unity or connection between the divided parts. So if the tertiary structure is a geometric 3D folding of moieties, it means like the subsections of the tertiary structure, which is basically the primary structure, which is a chain of amino acids, and the secondary structure, which is the beta sheets and the alpha helices. So it is the folding of moieties due to side chain interactions. So the side chains are interacting with each other to form an entire functional globular or structural protein. So what is the difference between globular and structural proteins? I think globular proteins are the ones that are secreting something. What are globular proteins? Characterized by their roughly spherical or globular shape. They are folded into compact structures and are soluble in water due to their hydrophilic, water attracting residues that are typically on the protein's surface. These proteins play various essential roles in the body. Some serve as enzymes facilitating chemical reactions, others as transport proteins carrying molecules such as oxygen, like hemoglobin, or lipids through the bloodstream. Additionally, globular proteins can function as hormones, antibodies, or molecular chaperones that assist in the folding of other proteins. The distinctive feature of globular proteins is their complex three-dimensional structure, often comprising multiple levels of folding, including primary, secondary, and tertiary, and sometimes quaternary structures. The primary structure refers to the linear sequence of amino acids, while the higher order structures involve the folding interactions of the amino acid chain to form the compact globular shape. This compact structure is essential for their function, allowing them to interact specifically with other molecules in processes vital for life. So, yeah, the important thing is to know that tertiary structure is the geometric 3D folding of moieties due to side chain interactions to form an totally functional globular or structural protein. Some proteins require multiple polypeptide subunits, all with their own tertiary structure, and connect them together to make a protein with quaternary structure. So some proteins require multiple polypeptide subunits, like hemoglobin, as we read about last night, all with their own tertiary structure, and connect them together to make a protein with quaternary structure. So they have their own tertiary structure, right? So some proteins require multiple polypeptide subunits, all with their own tertiary structure, and connect them together to make a protein with quaternary structure. Types of protein structures: alpha helix, beta pleated sheets. Yeah, so if you connect the subunits together, and each of those subunits having tertiary structures, then you will be making a protein with quaternary structure. Prolines are usually at the end of beta sheets for the chain to make a 180 degrees turn. So usually at the end in the beta sheets you see prolines, and this is where you can see that prolines are giving the kinks in the beta sheets. And proline is usually the first residue at the very end of an alpha helix. It is rarely inside the helix because it would introduce a kink. So at the end of each alpha helix, you will see, at the very end of an alpha helix, you see proline as the first residue, because otherwise, if it's inside the helix, it will give it a kink, which makes it a beta sheets style secondary structure. Let's go to redox reactions. Reduction potential or electrical potential, same thing, refers to the propensity of a chemical species to be reduced, gain electrons. So we know Gare, right? Gare, Leo Gare. Leo stands for loss of electrons, is oxidation, and Gare, gain of electrons, reduction. So you can see here that Ce4 plus is gaining electron, so it is reduced. This is the reduction half reaction. And because iron here is losing electron, it is a oxidation half reaction, right? Now we also have oil rig. O stands for oxidation, is loss of electrons. Oh yeah, I like Leo Gehrer because I started with that. This mnemonic describes the direction of electron movement in redox reactions. If a molecule is losing electrons, getting more positive or less negative, it is said to be getting oxidized. So if a molecule is losing electrons, right, getting more positive or less negative, it is said to be getting oxidized. So when you are losing electrons, of course, you are getting more positive or less negative, and it means that losing electrons is equal to oxidation. You're getting oxidized, right? And if you are gaining electrons, you are more negative, so less positive, and the more negative you are getting reduced. And a reducing agent reduces another chemical species. So the agent is called a reducing agent because it is reducing another substance. And an oxidizing agent oxidizes another chemical species. Don't get tripped up on this. Yes, I agree. It is very important to know that the reducing agent is not reduced itself. It is an agent that reduces other substances. Okay, let's go through these. Oh, lots of ones. It means every single one of them is a concept. So what spectroscopy measures how much, where are we? Separation techniques, lab techniques. These ones are, I didn't study any of them profoundly, but I find them tricky somehow. So let's get over it. Let's tackle it. And we see here that what spectroscopy measures how much a substance absorbs light based on its excitation in response to UV radiation. So when you are absorbing light based on excitation, because resonance allows molecules to spread out energy. Okay, let me go for a second and I'll be back.